So Lab 2 is tissues of the body. The four broad categories we will know are epithelial, connective, nervous, and muscular. You'll have 7 epithelial, 9 connective to know, 1 basic nervous tissue we'll talk about now, and 3 different muscle tissues. Remember, organs are made of tissues, so we study organ systems. We look at break that down into organs, and then to understand how organs work, you have to understand how tissues work. And again, kind of cells in a way as well, if you remember our hierarchy of organization from last week. So again, the 4 basic types, they differ very much in their matrix, what makes the cell up, how well connected the cells are, all sorts of things. With regards to the matrix, you have fibrous proteins of various sorts and various amounts of what's referred to as ground substance. Typically, extracellular fluid is how you would probably think of it in biology. Embryonic tissues, I won't particularly ask on an exam. They might be extra credit, but I just thought I might throw it in here. But there are 3 original layers that everything else kind of just derived from: the ecto, endo, and mesoderms. Again, think of the suffixes and prefixes. Ecto is the outer layer, endo is the inner layer, and mesoderm is the middle layer. Again, I won't really ask those, but they might be extra credit. So to understand what you see as far as the sections and the diagrams, cartoon pictures, etc., of tissues, you got to understand how they're kind of fixed and sliced. So usually tissues are fixed in a block of some sort and then sliced on one orientation or another. And again, you can get different information depending on how you slice those tissues. So start thinking about that when you are looking at diagrams of how that tissue might have been placed and then sliced. And we're looking at, basically we're looking through a slice when we look at tissue sections. Again, I encourage you to go to Google Images and type in a tissue name and see what comes up. All those photos should look similar in some respect or another, but they might look different especially in how they're stained. So tissues can look very different depending on how they're stained. Don't get used to the color of anything. Again, what you see depends on how you slice. So like looking at the next slide, how you see an egg would depend on where you slice it. If you slice it right down the middle, of course you got the nice fat diameter of the yolk. As you get closer and closer to the edge, the diameter of that yolk will get smaller and eventually disappear, and when you get that edge layer, that's purely albumin. So again, what you see depends on where that slice was taken of that full 3-dimensional object. Again, look at the macaroni slice. If you take the dashed lines and follow those down, if you took a slice at that level and looked at it, that would be what you'd see. Either just 2 holes if you're at the ends of the macaroni, or if you're partway in the bend, you might see part of that complex, or if you're just at the end, you might not even realize there's a tube there. Likewise, was showing you the 3-dimensional, kind of like a coiled tube. If you think of sweat glands, when we look at our skin slides next week, the sweat glands might not really be continuous with each other. When they go in and out of the plane of the section, it may not look like one continuous piece. So just keep that in mind when you're viewing these. Again, tissue is usually cut in just like our planes of the body, longitudinal section or cross section, and oblique section, again, is anything not 90 degrees or 180, somewhere between the two. And again, what you see depends on how that is sectioned. Of course, if you section a bone or a cylinder longitudinally, it would look very different than it would if you section it cross section or transversely. Again, oblique sections usually aren't aimed for, but there could be times when they're pertinent. Smears, what you usually see is with blood, liquid tissues, and some very soft tissues. You can sometimes see what's referred to as a spread, especially when you look at your areolar tissues. So again, epithelial tissue, I'll let you review these on your own. You can pause this, but I'm going to move on to the next slide. Epithelial tissue is sheets, all right, cells and lots of cell-to-cell contact. There is very little to no matrix. There's usually a basement membrane that covers one side of this sheet of cells. These cells could be, the sheet could be one cell thick or multiple cells thick. It just varies. There is an upper surface that's usually exposed to the environment or an internal space of the body. Remember, we are a tube, so what is in that tube is basically external environment to us. The breath that we swallow or food that we swallow or the breath that we breathe, that is basically external environment to us. So the same type of protection or surface-wise that we face outside of our body also faces the gastrointestinal tube within us, or many other tubes as well. Epithelia does not have blood vessels. It gets everything nutrient-wise from diffusion from tissues next to it. So functions of epithelia, think protection, chemical secretions, a lot of glands or epithelia, excreting waste and selectively filtering things. If you think that the single cell layer epithelia is a sheet, so it allows for to basically serve as a selectively permeable membrane. So certain things can pass through that sheet, certain things cannot. Again, very close cell-to-cell contact. Basement membrane is layer of connective tissue underneath the epithelial cells, kind of holding them together. Again, the cells themselves have a basal surface, which faces the basement membrane, and an apical, or sometimes referred to as free surface, which is the top surface that either, if you're thinking about skin, it faces the outside, or if you're facing a lumen of a tube or something like that, that this cell is lining, that is the apical or free surface. So epithelia is named by whether it is simple or stratified. It's kind of, you think of that as a first name. So the simple epithelia contains one layer, and it is named by the shape of the cell. So we have simple squamous, simple cuboidal, and simple columnar. When we look at the stratified epithelia, that is, think strata as layers. So we're looking at more than one layer of cells, from two cells to dozens, and that's named by the shape of the cells at the apical surface. So this indicates that cells can change shape as they move through through these layers of stratified epithelia. And cells will move through layers. They don't stay stagnant in one layer. They start in one layer, and as they divide and grow, they progress through the various layers of stratified epithelia. So again, with epithelia, the class is based on layers. You have simple, which is a single layer. Pseudostratified, columnar, appears to be stratified because the nuclei are not very lined up like they are in simple. All the cells in the pseudostratified attach to the basal membrane, but they don't all have an apical surface, but there is just one layer there. It's just what they're considered pseudostratified because it's falsely stratified, versus the stratified, which you see is you can see there's various layers of cells as they move from the basal to the apical surface. Also, you can see the cells change shape as they move from the basal to the apical surface as well. Again, with the shape, you're seeing kind of on the edge and kind of a tilted each one. With the squamous is very flat. Think like a fried egg, basically, if you're looking at that egg on end versus like on top of it. Cuboidal, don't think roughly square, but cuboidals are about as tall as they are wide. They may be hexagonal or kind of roundish in shape, depending on what's around them. They kind of fill the gaps in, but they're about as wide as they are tall. Columnar are 2 to 3 times taller than they are wide, and again, the placement of the nucleus is sometimes an indicator as to what you're looking at in these cells. Usually the nucleus is much more closer to the basal surface than the apical surface. But those are your epithelia based on layer numbers and shape. So again, the first four we need to know are simple, simple squamous, simple cuboidal, simple columnar, as well as pseudostratified columnar. Again, pseudostratified appears stratified, but it is really a single layer. One of the key features you can see often with pseudostratified is goblet cells. Oftentimes pseudostratified columnar is in areas that are producing mucus, and goblet cells secrete mucus, so they're often found with those. So a simple squamous epithelia, you're looking at a single layer of cells. Again, they permit diffusion from one side of the cell to the other. Remember, epithelia acts as a semi-permeable barrier. So those cells, they don't allow things to pass between cells, but the epithelia allow things to pass through cells, and as so they regulate what passes in and out. Again, the simple squamous are found within the glomeruli of the kidney. A good example is the alveoli within the lungs, endothelia, the capillaries, things like that. So simple squamous epithelia here, you see is that very top row that's highlighted in upper left side, that all those cells kind of end to end are the simple squamous epithelia. Simple cuboidal, again, the cells are about as tall as they are wide. Here we're looking at tubules of the kidney. And again, think back to your sections. We're looking at longitudinal and cross-sectional tubules there. You have the cross-sectional tubules that are circular in appearance, and the long two straight stretches of cells with the empty lumen between them is a good longitudinal section right along the length of a kidney tubule, showing that those cells do have a basal side and an apical side, apical or free side, again facing the lumen or the hole. Secretion, absorption, moving things from one side of the cell to the other side. Again, very much similar to what we saw before, but you should be able to recognize simple cuboidal. I think they're very indicative type of cell. Simple columnar epithelia, again, the nuclei pretty much line up. They're about the same spots in the cell. You can have goblet cells in simple columnar epithelia. A lot of simple columnar epithelia is associated with the digestive tract, and you're secreting mucus and stuff into the digestive tract. Now, what you see with simple columnar epithelia is what they call the brush border is microvilli. Now, remember, microvilli is different from cilia. Pseudostratified epithelia, pseudostratified columnar is ciliated, but oftentimes simple columnar epithelia, again found in the digestive tract, has microvilli. Microvilli are finger-like projections of the cell membrane of the cell, and that is to increase surface area for absorption into the cell. Thinking increasing surface area to absorb nutrients within the digestive tract. Again, you often see simple columnar with goblet cells as well as the pseudostratified. You'll see with goblet cells as well. So again, pseudostratified epithelia, you have cilia, and the job of cilia is different from microvilli. The job of cilia is to move mucus across the surface of cells. So think of the respiratory tract, and you're constantly moving mucus up to be expectorated and kind of spit out. A good example of this is the smoker's cough. When smokers smoke, it paralyzes the cilia, and when they don't smoke overnight, the cilia are active and they move mucus up, and the first thing when the smoker gets up in the morning, they're hacking and coughing because their cilia have been working overtime all night long. But that's, again, just know the difference between cilia and microvilli. I think it's very important to know the difference between the two and understand how they're just functionally different. Again, the nuclei in pseudostratified epithelia all over the place. It appears to be multi-layered, but really it's a single cell layer. Stratified epithelia, again, anything with layers. It can be from 2 to dozens of layers thick. We're only going to look at 2 here. Three, sorry. 2 stratified squamous, a keratinized and a non-keratinized version, and then a transitional epithelia. We will not look at stratified cuboidal or stratified columnar. They do exist, but they're rare, and we're just not going to look at them. Again, stratified squamous or stratified epithelia. The deepest layers continuously undergoing mitosis. So if you think about your skin is constantly being replaced because the deep layers are constantly dividing and pushing older cells out. As the cells move from the basal surface to the apical surface of the stratified epithelia, they age. So the surface of our skin is the oldest. The youngest cells are deep in that next to that basal layer. So we have keratinized and non-keratinized. Keratinized is on the skin, has a lot of keratin in these outer layers of cells. You have a thick, dead layer of outer cells, and non-keratinized lacks that. The difference functionally, you think non-keratin or keratinized is our skin. Non-keratinized is the lining of the mouth, vagina, and anus. These mucous membranes where you still need, abrasion protection. But you don't need the waterproofing. It's a moist membrane. So with epithelium, what you're looking at here is actually a skin slide. So this is epidermis, which is the epithelial layer, stratified squamous epithelia, and a little bit of dermis. On the right side of each of these photo and/or cartoon, the light pink, which is areolar tissue, that is the dermis, that is the deeper layer, and they kind of look like fingers interdigitating back and forth. But the kind of dark purple staining cells and that dark granular kind of line, that really dark purple line that you see, those are cells of the stratified squamous epithelia. As you see those cells go from that very dark purple line to a kind of a light staining area where you see it at the surface, it's dark purple. That is your free surface. That is the surface of our skin, and that is the keratinized cells. Now, as the cells go from that very, very dark purple layer up to the whitish layer, they lose their nuclei, and they basically replace all their organelles with keratin, a very tough waterproof protein. And that's partly what causes that part of the slide to tear. It's very waterproof, and those samples don't dehydrate well when you go to prep them. But again, you're looking at a keratinized, very thick layer of dead cells. You have no nuclei in those cells, but if you look closer to other cells, you will see nuclei. So here you see the epidermis, that is our stratified squamous epithelia, and you can see the kind of the bluish labeled dermis below. The stratum corneum, if you see down the bottom right, that is the layer that I was referring to. Those are all dead cells full of keratin. The last layer of cells you can call living is the stratum granulosum, and the cells start from the stratum basale and work their way up. We'll talk about that next week with skin, but it's worth mentioning now. So the stratum basale layers are the youngest cells, and stratum corneum, obviously the oldest cells that have died and everything been replaced with keratin. So in non-keratinized skin, you do not see the stratum corneum keratinized layer. So if you look at these cells of a non-keratinized stratified squamous epithelia from, again, mucous membranes, you see nuclei in cells all the way up to the surface. So again, if you look at the cells all the way at the apical surface, the top of this photo, you see they all have nuclei. You see they all have little teeny tiny dots that are stained. Those are the nuclei of the cell. So they're saying they're living cells all the way to the surface. So that's why a cheek swab is a good place to get a DNA sample from somebody. So if you think about things that get DNA samples, a cheek swab is an easy thing to do because those cells on the surface are very much alive. Transitional epithelia you see in the urinary tract, and it is kind of nondescript in a way. It looks like a bunch of kind of simple stratified cuboid cells, but it stretches, and you don't typically see it in slides under tension, but if these cells were to be stretched, they would thin out, and they would change shape as they kind of stretch. But transitional epithelia, again, you always see associated with the urinary tract areas where epithelial layer has to stretch a little bit in order to accommodate an influx of fluid. So transitional epithelia, again, you see there as it's lining the bladder, it kind of looks like the waistband on your elastic pants when you're not in them. It's kind of back and forth. But when it stretches out, that becomes a straight line. So connective tissue, again, I'll let you go over these kind of on your own. I'm just going to move along to the tissues themselves. So highly variable vascularity. We go from cartilage, which has no blood vessels at all, to loose connective tissue like areolar, and that's very, very vascular. So it varies. The big thing with connective tissue is all about the matrix, where epithelial tissue was just cells that were closely adhered to each other. We looked at the shape and the number of layers. Now we're looking at mostly the matrix, the fibers between cells in connective tissue. That's really what defines one connective tissue versus another. When you think of functions of connective tissue, they're very widely from support and kind of holding things together. When you think of tendons, ligaments, bones, immune protection, when you think of reticular tissue, that is the found in the spleen and the lymph nodes, and again, storage of energy in adipose tissue. Heat production will save for more muscle. I'm not looking at brown fat. We're not separating the different types of fat. We're just looking at adipose, or transport blood is a good one too. I mean, so many things are transported in blood. To really understand connective tissue, it helps to kind of understand the cells that you kind of will see in there. You may talk more about these in lecture, but I just want to mention briefly, we have fibroblasts, which produce the fibers of the matrix. I think collagen and elastin, those are two big fibers. We also have reticular fibers, which are a collagen, but it's a shorter branched collagen. But collagen, as we see in dense regular connective tissue, think of tendons, very strong ropes. The other is elastic fibers or elastin, which is very much like a rubber band. It will stretch out to a degree and then recoil back to its original length. Macrophages are immune cells, literally big eaters. They come from monocytes. When cells monocytes leave the blood and roam through the tissues, they're known as macrophages. The other leukocytes that do that are neutrophils as well, and lymphocytes, the B cells. They'll roam through the tissues as well, through the interstitial fluid. When they come into contact with a foreign agent and start to synthesize antibodies, they're known as plasma cells. So the B cells change names once they become activated, and that's basically what vaccines do, is they target B cells to create a population of plasma cells that are again synthesizing antibodies against that viral protein. Mast cells are basically basophils that have left the blood and roamed free, and they are secreting heparin and histamine to help regulate the immune response as well within tissues. And adipocytes, again, we'll look at adipose as a tissue, but the adipocytes are the cells that store triglycerides and fats, and you see adipocytes just in all sorts of different tissues, just kind of spread about. So I mentioned already the fibers of connective tissue are collagen, reticular fibers, and elastic fibers. Again, just kind of know some properties of those. Collagen is very tough, rope-like. Elastic fibers is like a rubber band. And reticular fibers form a network or a framework. They are much. They make little rooms in the organs that they're in. You might cover this a little more in lecture, but I'm not really going to cover the ground substance. But if you want to know more, I left a slide or two and it mentions it. With connective tissue, you have different ways of kind of separating them out, but loose connective tissue versus dense connective tissue separates out the kind of tissue, connective tissue proper, I guess, if you will. We have areolar, reticular, and adipose as our loose, and dense regular connective tissue as our dense. We will not look at dense irregular, and there are also other ways of dividing this up as well. So just be aware, not all text agree 100%. Areolar tissue, again, all 6 cell types. You see areolar in a lot of different places, packed under epithelia. This is a lot of times the basement membrane in epithelia. And when you look at their skin, the stratified squamous epithelia areolar tissue is in our dermis. So it's under a lot of epithelial tissue. Again, very loose, open, a lot of fibers oriented in different directions. You can sell various cell nuclei. Don't think like you're going to differentiate one versus the other right now, but just recognize those as various cells within the areolar tissue. Again, be able to recognize the areolar. One thing I often recommend to students is going to Google images and typing in the various tissue name types and trying to, identify those from Google Images. Reticular tissue, again, forms a framework within the spleen and lymph nodes. Thymus for immune cells to reside within the spleen. It helps to kind of break up old and decrepit red blood cells, but it's a framework for our immune system. Reticular tissue, those fibers are collagen fibers. Reticular fibers are just branched collagen fibers instead of like the dense regular where you have these long fibers of collagen. These are more short and branched fibers. Adipose tissue, again, large vacuoles of fat define these cells. The nuclei and the contents of the cells are pushed to the periphery. The fat usually does not take up stain, so you're left with looking like lace, this open area where the fat vacuole was, and the contents of the cell picking up stain on the periphery of that vacuole. Again, adipose tissues, you'll see that spread out all throughout the body in different areas. You can see the occasional nuclei of the adipocytes between the large vacuoles here and there. Dense regular connective tissue, again, parallel collagen fibers. Sometimes they'll pick up a bit of a wave to them in some areas, but think tendons, ligaments, things that are very strong. It's flexible, but it's not stretched. It's not something that's very well stretched. If it does stretch, it tears. So that's dense regular connective tissue. Some students have referred to it as pink wavy bacon before. Again, doesn't always have to be pink. Sometimes it can be blue. Cartilage, we're going to have three basic types, but to kind of understand cartilage and bone, we'll mention this a little bit with each, I think, but you have cells of each. Chondrus refers to cartilage. A blast is a cell that is creating something. So a chondroblast is creating the cartilage matrix around it. Basically, with cartilage and bone, the cells trap themselves in the matrix that they create around them, either cartilage or bone. So a chondroblast creates cartilage until it becomes a chondrocyte, and then it stops creating cartilage and just sits there in its little lacunae. The lacunae is the cavity or hole where that cell sits and spends the rest of its life. So you have lacunae within bone as well. There's no blood vessels in cartilage, so it heals very slowly. It is reliant upon diffusion, and cartilage is one of those things that the more you use it, actually the healthier it is for it. It is good for cartilage to be used when you step and release and you put pressure on cartilage and you release. It's kind of like squeezing a sponge and releasing a sponge. When you release a sponge, remember it sucks up the water around it, and that's kind of like what the cartilage does. It sucks up the nutrient-rich extra fluid around it, extracellular fluid around it. So it's good to use the cartilage, keep it in use. So hyaline cartilage is articular cartilage, and the background substance, there's no fibers in it. So the hyaline has a very glassy or clear background to it. You can see where the chondrocytes, the cells, sometimes where they've just divided, just almost paired up in a way as they're in the little lacunae. Again, the holes of the lacunae, the dark spots of the little cells, the chondrocytes, but there's no fibers in that background material. Hyaline cartilage is often known as articular cartilage. You see that trachea and costal cartilage, the end of long bones, and that's what our fetal cartilage, our fetal skeleton started off as. Our skeleton started off as a cartilage mold, and eventually that cartilage, that hyaline cartilage, became turned to bone, became ossified. So again, very clear extracellular matrix outside those lacunae. No fibers whatsoever in hyaline cartilage. In elastic cartilage, you will have elastic fibers. So elastic fibers usually stain kind of a dark color, dark purple. You don't see that quite so well in this diagram, but you look at this one and you definitely do. So elastic cartilage, again, you see in your ear and epiglottis, the external ear. So you can squeeze your external ear in every direction and it'll pop back into shape. But you see those same type of lacunae, the holes with the cells inside, but you also see the elastic fibers, as dark staining fibers in the background. So that's your elastic cartilage. Fibrocartilage has collagen, all right, bundles of collagen. So you'll see kind of a pink staining collagen as you see in this one. So you see the lacunae and little holes, and you can kind of tell how some of them are kind of running parallel to these collagen fibers. You see that in the pubic symphysis, the meniscus, the knees, and intervertebral discs, places where you need that cartilage to be very, very tough, a little more tough than regular hyaline cartilage. So in this diagram, you don't really see the collagen fibers stained so well. I guess you can see a little bit of the wave to the fibers in certain areas. It's an interesting photo. I've never really seen this one before, but you should see where there's a little bit of the wave to it, where you get the kind of the wave of the collagen fibers. But there are other diagrams out there where we can see these just a little bit better. So the two types of bone, we will only look at compact bone. We'll look at the structure of that. So compact bone, it is arranged in cylinders that surround a central canal that contains a blood vessel and life. Around the central canal are lamellae or layers. These are concentric layers that form the osteon. Between the lamellae are little holes known as lacunae where the osteocytes live. Between the lacunae, kind of radially oriented, are canaliculi, where the cell processes of each of these osteocytes reaches through and touches and is in contact with other osteocytes around it. So again, we'll visit the structure of bone in a little more detail, but some of these terms should be familiar to you. So again, with compact bone, you've got the central canal, the osteon. You see they're labeled; those are arranged around the central canal, and then the lamellae are the layers, the onion layers within the osteon. Blood is our only fluid. Connective tissue transports dissolved substances, gases, hormones, things like that, and also formed elements or cells. The erythrocytes are the red blood cells that transport oxygen. Leukocytes, leukos meaning white, are the white blood cells that are part of our immune system, and the platelets are fragments that help to create clots. We'll look at blood in a little more detail later. Again, blood is where you see a smear. Liquid is just kind of spread out. There's really no staining. Sometimes there's staining for the leukocytes, but there's really no staining for the erythrocytes. They're already pigmented. So again, nervous and muscle tissue, they're both excitable, which is what kind of makes them very similar. They have a membrane potential, which is you'll talk about in lecture later on. But basically, they send an electrical signal from one cell to another, and when a nervous cell sends that signal to a muscle cell, that muscle cell contracts in response to that. So nervous tissue is made mostly of neurons, such as the large stellate or star-shaped cell with large appendages. The neuroglia or glial cells are smaller support cells, and you'll just see the nuclei of those cells. You don't typically see neuroglia in a neuron smear. So again, with the neuron, the parts, we'll look more in detail at that later, but they have an axon, which carries the signal away to other cells. Dendrites are short and they carry the signal to that cell, and the soma, or the body, is sometimes referred to as a neurosoma, is the body of the axon, or the neuron. So here you've got two large neurons and the various neuroglia nuclei you see around them. So with muscle tissue, again, these cells contract. All they can do is shorten. And you've got three types: skeletal, cardiac, and smooth. Skeletal muscle is found within the skeletal muscles of our body. It is under voluntary control. We think about it, and the muscle responds, contracts in response to that. It has striations or stripes, which are the sarcomeres, the small contractile units, and it is multinucleated. Mature skeletal muscle cells are created from multiple cells that fuse together, each retaining their individual nuclei. So skeletal muscle has the large striations, multiple nuclei kind of pushed to the periphery. Cardiac muscle has striations, but it is under involuntary control. We do not think about it. Only found within the heart. It has striations, but these cells are singly nucleated. They are rather short compared to skeletal muscle cells. They are branched, and the cells have this interconnection called an intercalated disc that joins the cardiomyocytes together, and it allows that electrical signal to be transmitted readily from one cell to another. So cardiac muscle, again, has the intercalated discs and is branched. These are also single nucleus per cell with cardiac muscle. So with smooth muscle, you have this short kind of torpedo-shaped cell. They lack striations. They contract in a kind of a different mechanism. They're also under involuntary control, so they're non-striated, involuntary, and smooth muscle makes up the gut, basically, lines the intestines, small glands, things like that. So you don't think about digestion, but it occurs through the smooth muscle. And so the smooth muscle you can see in both a transverse and a longitudinal section. And there you see that the transverse section, you've kind of cut those muscles, those smooth muscle fibers on end. Or you see below with the longitudinal, you see those fibers kind of run left to right. Up above in the transverse section, they would run in and out of the plane, like perpendicular to the plane of the screen there. Individually, again, they're very what they refer to as fusiform or kind of torpedo shape. You think a typical muscle is a fusiform shape, it's wide in the middle and tapers off to either end. And that's what you see with a smooth muscle.